All right, everyone. Well, we'll get started now. We've given enough people time to kind of get involved and get settled in. People can join in as we get going. Obviously, as the main slide right here says, we are here for Mr. Shaman Patel. He's one of our master's students here at the Halmos College of Arts and Sciences. He's going to be working, obviously, from the title right there, in the Caribbean Reef-Building coral work, and he's going to have a great talk for us. He's already given me a little brief rundown of the presentation. So I'm really looking forward to hearing more about it myself as well. Shaman comes from a really diverse background, especially in the research field, includes experience with, lab settings, working with genetics and behavior. He's got field experience with both terrestrial and marine ecologies. He actually briefly worked with the Navy in their forestry and fisheries technician in the Pacific Northwest before coming to NOVA to pursue a master's in marine sciences. Since he's arrived at NOVA, he's been working with Dr. Swain's Evolution Symbiosis Lab. He's going to be investigating tolerance with marine heat waves in the Caribbean reef building corals, which is what he's going to be doing this presentation on today for us, which is really exciting. His project aims to identify what the corals that are critical stability in the Caribbean and the reef ecosystems. We hope to inspire restorations and efforts to focus on the long-term resilience-based management techniques for his labs and all his research he's going to be doing for us while he's a student here. He spends his time, outside of courses and thesis work, in restaurants, apparently eating as much food as possible and tons of ice cream, hanging out with his friends. So Shaman, I will let you, I'm going to stop sharing my screen. Why don't you go ahead and share yours? We're excited to, listen to what you have prepared for us today.
Thanks for the intro, Brad. Appreciate that. All right, let's get this started.
And while he's getting that set up, we will be doing a Q&A session at the end. So feel free to type any questions you may have, and then we'll answer them, when he's done, or, hopefully Shaman can answer most of them. If you have any other questions, we'll do our best to answer them as well.
All right. Should be ready to go. Hello, everyone. Good evening. Thank you for attending today. My name is Shaman Patel, and I came to NSU to pursue a master's degree in marine science. I decided to pursue a career in academia after studying abroad in the Galapagos. In the Galapagos, I got a chance to take part in fieldwork for multiple research projects. I worked with finches, marine iguanas, hammerhead sharks, while I was abroad, and continued to pursue opportunities to research how we can best protect animals from the dangers of human activity when I returned to my undergrad university. As I became more and more involved in ecological research, the topic of climate change, and especially the topic of coral reefs, became a personal interest of mine, and I really wanted to incorporate that in my work moving forward. I've been working with Dr. Timothy Swain here at NSU on my thesis project since about fall 2020. We kind of started talking preliminary then, going through literature, trying to figure out exactly how to approach this topic back then. And this is still a work in progress that we hope to have completed by this coming December. Today, I'll be providing the rationale behind my thesis and how we plan to address concerns with climate change as well as coral restoration. Specifically, I will be discussing how we can use systems biology and computational tools to identify the most critical coral species that require conservation attention in the Caribbean waters. Unfortunately, I will be giving everyone some bad news today, but we will also be discussing some good news too. Some of this good news actually comes straight out of labs from NSU, which is pretty impressive. There's always going to be some boring news, unfortunately, so I hope you all can bear with me through that before we end on a more hopeful note. Before we get into the details of the project, I wanted to lay out some important background information on corals. Specifically, we were talking about stony hermatypic corals. So hermatypic corals, these are the corals that form hard calcium carbonate skeletons and are the major coral reef building species. These corals also host zooxanthellae, which are single-celled algae that live within the coral's tissue. Zooxanthellae, which will also be referred to as zoox or symbionts today, use the sun's energy to convert carbon dioxide to sugars and fats, which they use for themselves as well as provide for their coral hosts. So these coral and zoox have a symbiotic relationship that is vital for their survival. Disruption of this symbiosis can cause an increased chance of mortality and morbidity in the coral hosts. It's very dangerous when we lose this. Now moving on to the bad news. It's getting hot in here. So unfortunately, current models predict that by the year 2100, global temperatures will increase by an average of almost 5 degrees Celsius. This increased global temperature will contribute to the increased frequency as well as severity of marine heat waves, which are predicted to become an annual event by about the mid-21st century. So we're talking less than 30 years from now. We could be seeing devastating heat waves occurring throughout the Caribbean on a yearly basis. Corals respond to this thermal stress from marine heat waves by ejecting their zooxanthellae. So this disrupts their symbiotic relationship in a process called coral bleaching. When corals bleach, they are more susceptible to disease and death because they are no longer receiving the services provided by their symbionts. They just lose a major food source. Marine heat waves are a natural occurrence that corals have dealt with in the past, but the increasing frequency and the severity of these heat waves are proving to be too much for corals to handle. An IUCN report showed that hard coral coverage on Caribbean reefs has decreased from 50 to 16% between the years 1970 and 2012. This is a major, major loss of our hard coral coverage. It is 2022 now, 10 years since the end of the last comprehensive survey of reef coverage was done in the Caribbean. And since 2012, there have been 2 severe widespread bleaching events occurring in the years 2015 and 2017. The worst global bleaching and mortality event on record occurred during the period between 2014 and 2017, and the current status of coral coverage in the Caribbean is unknown. It is likely, in my own opinion and most opinions that I've heard, that hard coral coverage has since gone below the last reported value of 16% in the Caribbean. So the picture here on top is from Carey's Fort Reef, just southeast of Key Largo in Florida. This image was taken back in 1975. And as you look closely, you can see a few species of fish and a generally well-structured reef, and that there's variation in the topography of this along the sea floor. So we got some height, we got some depth, and this is important, right? So this topography, this variation creates habitats and niches for all the wildlife that call coral reefs their home. The image below is the same reef, but this photo was taken in 2014. So there's almost no fish. The reef is essentially flat and barren. So why do we care about these dying reefs? First of all, there's a major impact on wildlife as a result of losing coral reefs. So coral bleaching puts an estimated 800,000 species of reef fishes at risk from habitat loss. The Caribbean itself is home to roughly 1,300 species of bony shorefish. 13% of these are considered threatened, including 19% of our endemic species here. So if you're a scuba diver or you like to be out on boats here, you might have heard of the Atlantic Goliath grouper or seen them in person. They're pretty abundant here in South Florida. But this is one of the species that is endemic to this area and is at risk of being lost due to coral reef habitat loss. So the degradation of coral reef habitats was identified as a key threat to bony shorefish, and South Florida is home to one of the richest bony shorefish biodiversity, followed by Belize and the Bay of Honduras. But these areas are at risk as their coral reefs continue to degrade. The loss of coral reefs is greater than just reduced biodiversity in our oceans, right? We're not just losing wildlife. It means less food, less tourism, and less protection for us humans as well. Overall, it means a reduced quality of life. So loss of jobs, suffering coastal economies, as well as just a loss of general economic value. So the estimated global economic value of coral reefs is nearly 10 trillion US dollars per year. Reefs also protect an estimated 1.8 billion US dollars in damages in the United States annually, but the collapse of reefs in the United States would likely result in damages more than double that value. So they help protect against hurricanes as well as tidal damage near coastlines. So we would lose that protection if we lose our coral reefs. That was some pretty heavy stuff, right? Pretty depressing, but thankfully there is some good news coming, some of which does come straight from here, from labs here at NSU. So successful restoration of coral. There has been an increased use of active restoration strategies on coral reefs in the past couple of decades to help us mitigate the loss of coral reefs. Restoration projects have shown a 60 to 70% survival rate in restoring hard corals to reefs. And not only are these corals being successfully replaced in the wild, but they're also growing faster too. So the growth rate of microfragmented corals, which is the technique they use to plant them, is about 25 to 50 times faster than normal growth rates. A meta-analysis from 2020 also showed us that there are 229 species of coral that have received some restoration attention. So a pretty decent diversity, a pretty decent amount of corals are being given some conservation attention. Although it is no replacement for significant action regarding climate change, restoration is a valuable tool for helping ecosystems retain their function by replenishing the dwindling coral populations at our reefs. However, there are some gaps in restoration efforts that are worth addressing, right? This is a pretty young field and we're still learning more and more about how to do this properly every day. So a meta-analysis from 2020 found that 60% of restored corals are branching type corals, half of which of those are from a single genus, Acropora. Acropora are a fast-growing and rapidly declining genus and are therefore good candidates for preliminary research on restoration methods. However, they might not be the most critical genera for a Caribbean network. Acroporids are particularly sensitive to thermal stress and may not be able to build reefs successfully as temperatures continue to rise. In other words, just planting Acropora might not be a solution that can withstand the looming thermal crisis. So how do we know what coral is going to last? Well, some corals are more tolerant to thermal stress than others. It was found that the species of zooxanthellae hosted by a coral has a significant impact on the overall coral's ability to withstand heat waves. But how do we examine that in a way that's useful? That's where network science comes in. So network science can conceptualize the complex system of the coral zooxanthellae associations, and it provides a means for determining the patterns of resistance to thermal stress and ecosystem stability during bleaching events. Networks can do this by representing their components in the form of nodes. So in our case, we'll have corals and Symbiodiniaceae phylotypes will make up the nodes of our network. And then these nodes are connected to each other by links. These links are known as edges, and they're used to represent an observed interaction. For example, you can have a social network, right, where people would be the nodes in the network, or they're connected to each other by links that represent the strength of their connection. A weaker link in this network could look like two people who work in separate departments at work and maybe only speak at meetings or after work parties. A stronger link would look like two people who are connected and have been best friends throughout childhood. So on the screen here, we have an example of one of these weighted networks. If we look at the connection between A to C and A to B, we can see that they're assigned values 8 and 9, which is a bit higher than the other values that you see in the network. So these could be those best friends, right? They might have known each other since they were kids. However, A to E might be a connection that maybe is just a coworker who maybe person B knows as well. So measures of a network include centrality, clustering, and node strength. There are some other ones, but these are some simple ones that I'm going to talk about today. Centrality refers to how central to the overall network a node is. So if we look at point C on this graphic, we can look at node D there in the middle. That will be a pretty central node. It's kind of placed in between, around, and is mostly surrounded by all the other nodes. It has more connections to the other nodes than the ones out on the fringes do. Clustering is a measure of how closely nodes in a network form together. For example, you can have low clustering where nodes are almost equally connected to all other nodes, so everything is just equally balanced, or higher clustering where clear groupings form in a network. These groupings can be indicative of certain things, such as like phylogenetic relatedness, for example. And finally, node strength refers to the sum of all weighted links attached to a node. So nodes with high strength have many or strong connections to the other nodes that they are attached to. All nodes with weaker node strength are characterized by either less connections or weaker connections. How can network science help restoration? So Dr. Swain, my advisor, did some previous work to establish a taxon-specific bleaching response index. That's a mouthful, which is basically just a robust metric of intrinsic susceptibility of coral taxa to thermal stress. In other words, we have a measure for how tolerant corals are to heat. We just need to know what zooxanthellae they host to obtain their combined BRI value. These values can be used to make those weighted links I mentioned earlier. So instead of the strength of a connection one person has to another, in the example I just brought up, corals will be connected to zooxanthellae based on how well they withstand temperature as a combined unit. This is a useful linkage to have, as we can, for example, artificially simulate temperature increase in the network to reflect predictions based on global temperature models. As temperature rises, links will be removed and the network will change its overall structure. These changes can be quantified and are useful for identifying corals that can cause significant shifts in the characteristics of the network. So losing some core nodes, some core corals in a network might cause either multiple networks to form or different clustering to be evident. So to reiterate, because the unique traits of corals and zoc species combinations determine their vulnerability to thermal stress, the regional loss of coral species is potentially predictable. So we can figure out what corals we're going to lose as temperature increases. What is not yet predictable is how the loss of coral and their associated symbionts will affect the remaining symbiosis network. So loss of corals or zoc nodes in the symbiosis network may destabilize the remaining links and further endanger species that are thought to be more resilient to climate change. A complete network will provide a basis for how we can predict the way this loss of vulnerable coral zooxanthellae associations will affect the entire network. So how will we use network science? How can network science be applied to the issue of rising temperatures and restoration? We plan to develop a network model that represents all Caribbean hermatypic corals, that's those reef-building corals, and their associated symbionts. This network will be layered with metadata that includes population status, conservation status, phylogenetic relationship between the hosts and the symbionts, thermotolerance of hosts and the symbionts, as well as transmission mode of the symbionts. The proposed network model should consist of all 67 hermatypic corals. It's an ambitious goal to get every single coral accounted for. But the network will also include Brazilian and South Florida corals to reflect the most inclusive community of corals that could build reefs in the Caribbean region. This model will allow us to identify first the network critical coral symbiont associations, and second, which of these critical species are best adapted for surviving inevitable sea warming events. Understanding these things will allow us to answer the following questions. What corals or coral symbiont associations are most vital to network stability in the Caribbean? So the most highly connected nodes are typically responsible for maintaining the core of the network structure. Losing these nodes or these corals can cause shifts in the network that changes the overall structuring. For example, possible outcomes of losing these critical corals could lead to the severing of our network into two separate unconnected systems. Which of these associations can be protected by increasing conservation effort on the coral hosts? So this will be addressed by layering metadata for conservation and population status onto the network nodes. By doing this, we can pinpoint which corals are critical to stability, as well as the most at risk of extinction, or maybe they just need some more conservation attention to protect the system overall. And the final question, so how resilient and how robust is the Caribbean network to thermal stress? So this final question seeks to quantify the temperature threshold where the Caribbean network loses about 50% of its nodes. This would constitute a significant loss of the reef-building corals in the Caribbean and could have disastrous impacts on the services provided by our coral reefs. It is also crucial to know exactly how the Caribbean corals will respond to rising sea temperatures. Quick sip of water. And now for the boring part, how we plan to answer these questions. I've talked a bit about the network we will build. But in order to run the type of analyses that make the network useful, there are some other steps we have to take. We first need to know what corals exist in the Caribbean. And second, we need data on what zooxanthellae species are associated with the Caribbean corals. To find out what corals are in the Caribbean, I searched literature and coral databases for any stony corals known to host zooxanthellae that had a reference distribution in the Caribbean, within the area of our focus between Brazil and South Florida, and from Belize all the way out to Bermuda. The next step was to determine what types of zooxanthellae are associated with these corals. To find this out, we did a combination of two things. The first is continue with searching scientific literature for published coral symbiont associations. With the help of a couple of undergraduate students here at NSU, Mackenzie and Brady, if you're out there, shout out. We tracked down papers that mentioned zooxanthellae and the corals in the Caribbean we needed data for. We looked specifically for the ITS2 phylotype of zooxanthellae collected from coral fragments. The internal transcribed spacer 2, ITS2 region of nuclear ribosomal data is regarded as one of the best candidates for DNA barcodes because it has some characteristics valuable for our purposes, such as the availability of conserved regions for designing universal primers, the relative ease of its amplification, and sufficient variability to distinguish even closely related species. The ITS2 phylotype is therefore a good way to identify zooxanthellae species in coral samples. We searched literature until we got at least 10 coral symbiont association records for each of our corals. We exhausted the literature search and had sufficient data for 34 of the 67 Caribbean corals, or a bit over halfway already. We then proceeded to collect new samples in the Caribbean to help us bring us closer to the complete network. The field techniques were pretty simple. Scuba divers just collected some coral samples, they removed the fragments, and then sent us the samples here at NSU for DNA extraction. These fragments were then preserved after they were sent to us here at NSU, where we will be doing the molecular work to determine the ITS2 phylotype. We will most likely take a Sanger sequencing approach just because it's cheap, and then check against blast sequences to determine the phylotypes of the symbionts present in each sample. We're also investigating adding the SymPortal framework as another route for determining the symbiont profile type. Our bleaching response index is reliant on the phylotype data, so one way or the other, we will still get the identities of the symbionts so we can continue with our network analysis. We plan to include the complete ITS region in our sequencing, along with complete mitochondrial cytochrome B and chloroplast 23S partial sequences from the nuclear 28S and 18S regions will also be included. These regions were chosen specifically because they have been shown to be highly variable and are suitable for phylogenetic analysis. Although ITS2 is reliable on its own for distinguishing the symbiont species, the addition of the COB, the CP23S will provide supplementary data from which we can corroborate or potentially question the current practice of relying solely on ITS2 for determining the Symbiodiniaceae phylotypes. So sequence dissimilarity data for the CP23S domain 5 indicated that the region has significantly higher evolutionary rates than nuclear small subunit, large subunit, and ITS regions, and was used to provide the first independent evidence supporting the major clades of Symbiodiniaceae. This is likely gibberish to anyone who isn't familiar with this type of genetic work. So simply put, we will be extracting DNA from areas within the genomes of the zooxanthellae that have been proven to be reliable ways to distinguish one species from the other. Completing all this molecular work should bring our total coral species represented in the network to 38 out of our 67. Now, I know the equation on this slide can be scary at first. I darn near changed my thesis project when I first came across it. But thankfully, it's not only a fairly simple equation, it's also extremely useful for this project. Our network will be constructed using the iGraph package in R, and there will be two types of nodes, one for coral hosts and another to represent the symbiont phylotypes. These nodes will be connected by weighted links provided by the equation on this slide. This equation is the bleaching model. This bleaching model assigns values to the links between nodes based on a couple expectations. The first is that corals will bleach once temperatures exceed mean monthly maximum sea surface temperature. The second is that there is an upper thermal limit for coral symbiont association. So corals will be considered bleached once temperatures exceed the norm, and that corals will lose their symbiotic relationship with their zooxanthellae at a certain temperature. That's basically what this equation tells us. To break down this model further, T sub triple M represents that mean monthly maximum sea surface temperature. The host and symbiont tolerances are averaged. That's the tau symbiont plus tau host, to account for uncertainty of which partner is responsible for the breakdown of the symbiosis. T delta is the upper thermal limit for the mutualism interaction, and the average tolerance is multiplied by the upper thermal limit to give a measure of how much of the thermal limit a pair can tolerate. This model will be used in conjunction with a bunch of null networks to help us understand how the structure of the Caribbean network and the distribution of thermal tolerances might affect resistance to temperature stress. I'll go into further detail on the null networks on the next slide. The resistance of the Caribbean network will be determined by calculating the required temperature for the network to go from 10% to 90% bleached. So ecological robustness will be calculated using the seven link removal models that will each answer a slightly different component of robustness. So ecological robustness will be ultimately measured by finding the R50 values. The R50 value is just the number of nodes or links needed to be removed until only 50% of the original nodes are remaining in each of the removal models. So for the random links model, so links will be removed in random order and serve as a baseline for comparison for other link-based removal models. This is very similar to the random nodes, which does the exact same thing. Susceptible nodes and susceptible links are very similar. So links and or nodes will be removed based on the tolerances of the combined holobiont. So the holobiont refers to the combination of the zooxanthellae as well as coral in order from low to high tolerances of thermal stress. Holobiont tolerance will be calculated as the average tolerance of the coral host and the linked symbiont phylotype, with thermotolerance scores being pulled from recently standardized indices. That's that taxon BRI that Dr. Swain worked on. Bleaching, links will be removed based according to the bleaching model as described in the previous slide. Population status, nodes will be removed from high risk to low risk of extinction based on IUCN scores for coral species. Corals with insufficient population data will be left out of this model. And finally, conservation status, conservation attention. So coral nodes will be removed from low to high conservation status provided by EDGE. That's the evolutionary, distinct, and globally endangered scores. EDGE scores are created by combining scores for both unique evolutionary history for each species, as well as its global conservation status. These EDGE scores also include a measure for the conservation attention a species is receiving, and it ranges from good, medium, low, or very low attention. We will be layering all of this into our network model. So this project is still underway. We are currently working on finishing the molecular work, and I will be getting deep into the network analysis in the coming months. I have started the network coding, and I can show you what we have for the Caribbean so far. So here we have 32 Caribbean corals represented in a preliminary network. The red circles represent the coral hosts. The gray squares are the symbiont phylotypes. We already have some clear clustering of coral hosts, as you can see kind of on the left. There are also some corals more central to the network, and some, like the one way at the top, that are weakly associated with the rest of the network. Once all the molecular work is complete, I will add the data from those corals and a couple more that were recently fulfilled by literature research. And soon we will have 38 of the 67 corals represented in this network. So we're looking at about 50% of the total network in this study alone. 67 corals is an estimate of the coral species we have in the Caribbean. Unfortunately, as we mentioned earlier, there is a major time block from when the population status of corals in the Caribbean was updated. The latest IUCN data dates back to 2008, and there have been many major bleaching events since then. It is likely that there are less than 67 reef-building corals in the Caribbean, We're hopeful that this is not true and urge others to continue to gather symbiotic association data on coral to build our network to be as complete as possible. So I'm looking forward to working on the network analysis and finding some answers to our research questions. The final deliverables of this project will include one, a reference list of all hermit-typic corals in the Caribbean. Two, a short list of corals that hold associations vital to network stability. Three, a short list of network critical corals that require more conservation attention. Four, the tolerance measured by resilience and robustness of a network, of the Caribbean network, to thermal stress. And five, a plug and play tool for recreating this analysis for any ocean region that provides the deliverables 2 to 4 mentioned above. All genetic data collected during this project will be submitted to GenBank, and all data files and R/Python scripts, because we're working with 2 programming environments here, will also be made publicly available. Our network and analysis code will be packaged as a tool, so researchers globally can just get a list of corals that they want to look at for their reef, find out what zooxanthellae are associated with those corals, plug and play into an Excel file, and have the R and Python scripts run automatically. So they don't have to go through all the pain I have to learning all this stuff for the first time. And yeah, other people can recreate this elsewhere. Thanks everyone for tuning in. Now I'm ready for some questions or comments if you have any. Again, some in the chat. So the first 2 questions are one from my mom: what inspired you to go into this work? And the other one from Andrew Gebner: what made you choose marine biology? Good questions. So I started off in my undergrad kind of going the pre-med route. I ended up studying abroad in the Galapagos and realized that research is what I want to do for the rest of my life. I was involved with projects out there, waking up at 4 o'clock in the morning to go monitor birds for 3 hours in the Galapagonian heat. And I didn't mind a single second of it. So I knew I was in love with this type of work. Marine biology just kind of came to me as I continued working in the field of ecology and researching in the field of ecology. We're losing our corals, we're losing our fishes, and I think it's just where I need to be. Thank you, Megan. So Brett asked, when will it be possible to get the information on all 67 species of coral? It's possible as soon as someone has enough money and time to go do it. Colloquially, I've been talking to a few people that are familiar with Caribbean corals, and it seems like 3 or 4 of the corals on our list are already considered to be extinct in the wild. Not officially yet, because IUCN, report isn't out, but in their experience in the decades that they've been working here, they either haven't seen these corals, don't know anyone else who's seen these corals in the last 10, 15 years. So we might not have to get 67, we might only need to get about 60 at the end of the day. And we're getting pretty close to that already. So it's just time and money. Get some more grad students on it. So Andrew Gebner asked, what are your biggest challenges with completing your project? Great question, Andrew. The biggest challenge has got to be getting through all this coding. So there's a lot of modeling that we have to do, a lot of network analysis that needs to be done. And while I'm familiar with R and I'm familiar with Python, this is all very new applications of it for me. So learning that has got to be the biggest challenge. I've been very lucky. My mentor has been very helpful with teaching me all the biology and ecology and phylogenetic stuff that I need to know. But this is the piece that's more independent, and this is the one I've had to struggle with the most recently. Good question. So Gretchen has asked, in what you have seen so far in the research, as well as your own knowledge about biodiversity, what do you suspect you may see regarding your networks and the connectivity of these species to the ecosystem? Heck of a question, Gretchen. So it's difficult, right? Network analysis is a great way to model complex ecosystem interactions, but it doesn't take into account a lot of things, because modeling in itself is a very simplistic concept, right? The more simple a model is, it tends to be more powerful. The more complex you make it, the less applicable it is going forward. So this is a great, this is great information for us to know how corals will work with each other in terms of building a reef, how it's going to impact biodiversity, how it's going to impact the species of fish that we see, of sharks, etc. That has a bit more facets to it. And that's where understanding functional diversity, understanding exactly what fish associate with what corals for their habitats and reefs becomes very important for us to know. So more investigation on that, or incorporating previous investigations on that in a network analysis, will help build the model, while also accounting for those kind of larger ecosystem interactions that deal with overall biodiversity. Sweet question. Appreciate it, Chris. Brett has asked, what is the best way for the average person to help? Great question, Brett. I'm sure everyone has seen campaigns for skipping the straw and driving hybrids or electric cars. And those are all fantastic ways to get involved at a personal level in just being more sustainable and being more eco-friendly. But the biggest impact I think any one person can have is by being involved in policy and government as well. It's our major corporations, it's our industry, it's our infrastructure that's creating all the pollution, all the climate changing activity that's harming coral reefs. So being involved in politics, whether you serve on a local board or you vote in candidates who not only promise but actually follow out and carry through with creating policies and enforcing policies that promote sustainability, that's got to be the best way to be involved as an average person, at least in a way that has the biggest impact.
I think that probably just wraps up, most of the questions. Obviously, Shaman, thank you so much for, volunteering some time. I know you got a lot of work to do and you're obviously really busy. We see you running around everywhere you go over at the oceanographic facility. So we really do appreciate, you taking the time to do this. It was a fantastic presentation. If anybody does have further questions for him, please feel free to, reach out to the email that you have, and we'll see if we can't forward on some further questions from there. I hope everyone, enjoyed this presentation as much as I did. I know I'm looking forward to paying more attention to these type of things in the future. And I look forward to, everyone hopefully enjoy your upcoming weekend. Obviously think about this when you're out going to the beach or whatever you happen to be doing, but thank you once again for, attending and listening to Shaman talk about his research. It's been a lot. Thank you so much.
Thanks everyone. Thanks for the opportunity, Brett.